Glycogen structure and linkage types. Which of the following best describes the types of glycosidic bonds found in the glycogen molecule? A. Vata4 for branching, A116 for linear segments. B. A114 for branching, beta-116 for linear segments. C. B16 for both linear and branched areas. D. A14 for linear 6 at branch points. E. V14 for linear chains, 16 at branch points. Correct answer, D. A14 for linear chains, A16 at branch points. Explanation: glycogen is a branch polysaccharide made of glucose units. AV14, glycosidic bonds form the linear chains. At a 6, glycosidic bonds form branches approximately every 8 to 12 glucose residues. These linkages allow glycogen to be densely packed and rapidly mobilized. Why not the others? BEPA linkages are found in cellulose, not in glycogen. The types and orientations in A, B, C, and E are incorrect or reversed. Question 2. Glycogen use during muscle activity. Which of the following best describes the role of glycogen in skeletal muscle during exercise? A. It is broken down to glucose and released into the bloodstream. B. It is used to maintain blood glucose via gluconeogenesis. C. It is rapidly broken down to glucose 6-phosphate for local ATP production. D. It is converted to free fatty acids for sustained energy. E. It is inhibited during anaerobic conditions. Correct answer C. It is rapidly broken down to glucose 6-phosphate for local ATP production. Explanation: in skeletal muscle, glycogen is not used to regulate blood glucose. Muscle lacks glucose 6-phosphatase, so glycogen, glucose 6-phosphate, not free glucose, enters glycolysis for local ATP production, especially during exercise. Why not the others? A equals liver glycogen does this, not muscle. B equals gluconeogenesis is a hepatic process. D, glycogen is not converted to fatty acids. E equals glycogenolysis is activated during anaerobic activity. Would you like a mnemonic for glycogen?